Human immunodeficiency virus type 1, or HIV1, attacks the body's immune system by targeting specific cells that help the body fight off infections. Over time, the virus continues to destroy these cells, in turn weakening the body's immune system. Lack of treatment can reduce the number of these cells significantly, leading to acquired immunodeficiency syndrome, or AIDS, which is characterized by frequent severe illnesses. HIV1 is a retrovirus that attaches to the membrane of the targeted host cell. Upon attaching to the membrane, the retrovirus enters the cell and uses an enzyme, reverse transcriptase, to convert the viral RNA to DNA. The DNA is integrated into the host cell's DNA and allows the HIV1 retrovirus to replicate. HIV replicates leave the host cell to mature and spread throughout the body to continue the cycle. The HIV1 genome is comprised of 9 genes, all necessary for the formation of an infectious cell. One of these genes, the GAG gene, plays a pivotal role in the synthesis, transport, and assembly of the GAG polyprotein. This polyprotein is important because many of the structural proteins needed for a mature HIV1 particle are derived from it, including the nucleocapsid protein. The nucleocapsid protein is a small basic protein that plays a major role in the virus replication cycle, including RNA packaging and virus assembly, reverse transcriptase, and replication. The NC protein is able to act as a nucleic acid chaperone, meaning it has the ability to remodel nucleic acid structures to their most thermodynamically stable conformations. The NC protein contains a highly basic N-terminus, a short C-terminus, and 2 zinc binding domains connected by a peptide linker. Smaller proteins often require some sort of structural stabilization, which can often come in the form of metal ligand binding. The zinc binding domains, or zinc fingers, are comprised of the metal ion zinc 2+, and the 4 amino acid ligand, comprised of 3 cysteines and 1 histidine. In this molecule, the ligand is considered a chelator because it binds to the metal center through more than one donor group. As one multidentate ligand, the protein is able to bind to the zinc 2+ metal center through 4 donor atoms, corresponding to the 4 amino acids in the chelator, effectively replacing the water molecules of the hydrated zinc. The increase in molecules witnessed in the reaction of hydrated zinc with the multidentate ligand can be explained by the chelate effect, which results in an increase in entropy and an overall favorable reaction. This zinc finger domain provides a more thermodynamically stable complex than an analogous zinc complex with individual cysteine and histidine amino acids. The specificity of the ligands can be attributed to hard-soft acid-base theory. Metals and ligands are categorized based on their hard and soft characteristics. Overall, acids or bases characterized as hard typically have a higher charge density, and those that are soft display a lower charge density. In this theory, metals and ligands take on a like prefers like trend, where hard metals will prefer to bind to hard ligands and vice versa. The zinc ion, zinc 2+, is classified as a borderline acid. The presence of both cysteine and histidine residues give the NC metal binding site a mixed soft borderline character due to the presence of the soft thiol group on the cysteines and the borderline imidazole nitrogen of the histidine residue. Since like prefers to bind with like, there is a strong selectivity between the zinc 2+ ion and the cysteine and histidine ligands because of their similar soft acid-base characteristics. According to ligand field theory, metal complexes will prefer to adopt the configuration with the lowest ligand field stabilization energy, or LFSE. The value of LFSE corresponds to the balance or imbalance between the higher orbital and lower orbital electrons. In the case of zinc 2+, a D10 metal, both the high and low orbitals are filled, indicating a balance between the higher and lower orbital electrons. This balance corresponds to an LFSE value of 0, indicating that zinc 2+ has no LFSE preference for a specific geometry. With no preference for specific geometry, sterics become the major factor in determining the geometry. This also explains the selectivity of the zinc finger domain. Other metals that do not have a full D shell will have an imbalance between higher and lower orbital electrons. This will result in a negative LFSC value, which indicates that the metal will have a preference for a geometry, most likely octahedral. This phenomenon helps explain the selectivity for zinc 2+ over other metals which have specific preferences for non-tetrahedral geometries. Since zinc is a relatively small ion, more ligands bound to the ion will only produce unfavorable steric interactions. Understanding this, zinc will favor a 4-coordinate tetrahedral geometry over a 6-coordinate octahedral geometry to reduce steric stresses. This tetrahedral geometry provides a nice balance between the steric factors imposed by the ligands and the electrostatic factors which come from the ligand electron density that helps to stabilize the +2 charge on the zinc ion. Sterics are also the reason that the tetrahedral geometry is chosen over the 4-coordinate square planar geometry. Larger angles between the ligands, 109.5 degrees, in the tetrahedral geometry provide more favorable steric interactions than those encountered in a square planar geometry with 90-degree angles. Within the HIV1 nucleocapsid protein, as previously mentioned, there is an LFSC of 0. It would be expected that the molecule would be labile and thermodynamically unstable because the zinc 2+ ion lacks the stability of the lower orbital electrons. However, within this molecule, it is understood that the +2 charge of the zinc 2+ metal ion allows for strong electrostatic interactions with the negatively charged ligands, resulting in the metal complex adopting more inert qualities compared to a zinc 2+ metal complex with more neutral ligands. These principles of coordination chemistry help explain the pertinent role that the zinc finger motif plays in the life cycle of the HIV1 NC protein and the overall HIV1 retrovirus. Because these zinc fingers play such a major role in the life cycle of HIV1, much research has been done as of late to design zinc finger inhibitors that are able to disrupt the function of these zinc fingers in the HIV1 NC. Although much more research is needed, this could be a key player for combating HIV in the future.